Hello, today we are moving on from where we had stopped at, and today we will look at physiological processes. Now, in the previous lesson, we had learned that the cell membrane always allows the movement of materials in and out of the cell. Now, the processes which enables the cell to ensure that the materials are moving in and out are referred to as the physiological processes. Now, they include diffusion, osmosis, and active transport. But for today, we will look at diffusion. Diffusion. You'll find that particles in a gas or a liquid are always constantly moving freely, and as they move, they bump onto each other and they bounce off randomly. Now, when this occurs, you realize that particles always move from a region of higher concentration to a region of lower concentration, and this results in particles being evenly spread within the space that is available. For example, when you open a bottle containing perfume in a room, after some time you'll realize that the scent of that perfume will be actually evenly distributed within the room, and areas where we were not having that perfume, you will always feel that scent. And this is because the perfume will always be moving from a region of high concentration that is within the bottle to the other parts of the room where the concentration was low until it is evenly spread. Therefore, we can now define diffusion as a process by which molecules move from a region of high concentration to a region of low concentration. And now we move on to practical activity 1, and in this case we will try to carry out, or we will just discuss an experiment that tries to demonstrate diffusion using potassium manganate 7, or what is commonly known as permanganate. Now, what you require for this experiment include the following. You will require potassium permanganate 7 crystals. 2, you will require glass tubing. 3, you will also require 100 cubic centimeters beaker and water. Now, the procedure includes: 1, you hold a glass tubing vertically in the beaker such that in one end you place your finger, and the other end should be having the crystal of potassium permanganate. Then you carefully drop the crystal of potassium permanganate into the beaker. Then after that, you have fill the beaker with the water. Then after filling it with the water, you carefully withdraw the glass tubing so that you ensure that the crystals of the permanganate 7 they remain undispersed or undisturbed at the bottom of the beaker. Then you will leave it for a period of around 15 minutes. Then you will observe the changes. Then after that, you record them. Then lastly, you'll be required to make an explanation of what you have observed. So from the demonstration that we have here, you will realize that after a period of around 15 minutes, the potassium permanganate 7, which is always purple in color, will always spread throughout the water evenly. And at the end of the day, you will realize that the water will turn purple. Now, the explanation for this is that the crystals of the potassium permanganate 7, it has higher concentration of potassium manganate 7 molecules. So they will always move from where they are highly concentrated to other parts of the beaker where their concentration is low. So by that, you realize that these crystals of permanganate 7, they move by the process known as diffusion. And when you talk about these physiological processes, it is wrong to talk about through. You know, you move through the window or through the door, but for a process, it is by. So many students make a mistake of talking about through, but the right one should be by. So the potassium manganate 7, it will always move from where the molecules of potassium manganate 7, they will always move from where they are highly concentrated to where they are lowly concentrated by diffusion. So you look at step 1, we are placing in the crystal of potassium permanganate 7. Then we fill it with water, then we withdraw the glass tubing. So you can realize that in the third diagram, we don't have the glass tubing. But if you look at the crystal of potassium manganate 7, you realize that the molecules are moving from where they are highly concentrated. It is breaking off from the potassium manganate crystal and it is moving to other parts of the liquid until that time we have the potassium manganate, the molecules of the potassium manganate 7 being evenly distributed within the beaker that we have around here. Now we move on to the next thing, and we are introducing another term, and now this term we refer to it as concentration gradient. Now, how does concentration gradient comes about? Now, when we have the difference between a low concentration region and a high concentration region, then we will always refer to it as concentration gradient. So you find that when we define concentration gradient, it is the difference of concentration of molecules between the regions of low concentration and the region of high concentration. Now, diffusion as a process, it is always a passive process, and it cannot take place whenever there is no concentration gradient. So it is a process which only takes place when there is concentration gradient. Now we are moving on to the next practical activity, and we will try to explain or we'll try to demonstrate diffusion using whisking tubing. Now for us to carry out these experiments, we need the following. Whisking tubing, we need a whisking tubing of around 8 centimeters long, starch solution, dilute iodine solution in a 100 millimeter beaker. We need a thread, a glass rod, dropper, and water in a beaker. Procedure: 1. Tie one end of the whisking tubing using the thread provided. So what you'll do, you'll first tie one end, then the next thing, you will pour starch solution into the whisking tubing about 3/4 full, then you tie the other end. So if you look at this whisking tubing, this other end which is tied, then after that you pour in the starch solution, then you tie the other end. Then after that, you ensure that you tied it tightly such that the whisking tubing is not leaking. Then you rinse it with the water. After rinsing it in water, you immerse the whisking tubing into the beaker which is containing the iodine solution. Another step, you allow the setup to stand for about 20 minutes. Then after 20 minutes, you should be in a position, you should remove the whisking tubing from the beaker and you record the observation. Then lastly, you should account for the observation that is observed. Now we move on to the next thing, and now we want to make a discussion of what will happen at a period after a period of 20 minutes. So you will come to realize that after a period of 20 minutes, you will realize that the contents of the whisking tubing will always turn blue-black. Now the question you should ask yourself, when we were looking at when we were looking at iodine solution, remember the color was the color of iodine was brown, but the starch solution it was somehow colorless. Now you realize that after some time the starch solution within the whisking tubing, it will turn blue-black. Now that is what we want to explain. Why is it that the color of the contents of the whisking tubing turned blue-black while the iodine solution retained its brown color? Now this is because the wall of the whisking tubing, it is always semi-permeable. And when we talk about the whisking tubing wall being semi-permeable, it means that it only allows molecules of small particles to pass through. So you'll find that only iodine solution molecules could pass into the whisking tubing, and the starch solution could not have its molecule passing out of the whisking tubing. So you find that a reaction occurred whereby the iodine molecules which passed into the whisking tubing reacted with the starch solution to form a blue-black solution. So what is going on here is that, or what you should know is that the iodine molecules, because they have small molecules, they moved into the whisking tubing through the semi-permeable membrane by a process known as diffusion. So it is the iodine solution are moving from where they are highly concentrated to into the whisking tubing where they are lowly concentrated through the semi-permeable wall. We move on to the next stage, and now we want to look at factors that affect the rate of diffusion. Now the first factor that we will look at is thickness of tissue. So you find that the thicker the tissue, the slower the rate of diffusion. Why? When the tissues are thick, then the diffusing molecules, they have to travel for quite a longer distance as compared to when we have the tissue being thin. So when the tissue is thin, the diffusing molecules cover a shorter distance. Hence, the rate of diffusion will be higher. Another factor is size of the molecules. So you find that small and light molecules are diffused faster than heavy molecules, which are always, larger molecules are heavy, so they move at quite a slower pace. So the larger the molecule, the heavier it is, so it diffuses at a slower rate. Another factor is temperature. So you find that an increase in temperature will always increase the energy content of the diffusing material. It increases the kinetic energy or the energy content of the diffusing materials. Hence, an increase in temperature will always increase the rate of diffusion, while a decrease in temperature will always reduce the rate of diffusion. Why? When the temperature is decreased, the kinetic energy of the molecules will always be lowered, and hence they will diffuse at a slower rate. Another factor is type of medium, and here we have diffusion always taking place in gases and liquids and not in solids. But if we compare the rate of diffusion in gases and in liquids, we'll realize that diffusion is higher, or the rate of diffusion is higher in gases than in liquids. Another factor is diffusion gradient. So you find that the greater the diffusion gradient between 2 points, the higher the rate of diffusion. Therefore, increasing the concentration of diffusing molecules at one point will always increase the rate of diffusion. Another factor, we have surface area. So you find that the larger the surface area, the higher the rate of diffusion. And this one is commonly seen in lower animals like amoeba. That is the process which is always used by this lower organism to carry out gaseous exchange and other processes. So the larger the surface area, the higher the rate of diffusion, because most of the surface of this organism is always exposed to the environment. Lastly, the last factor, we have surface area to volume ratio. So the rate of diffusion in and out of the body always depends on the surface area to volume ratio. Now you realize that the higher the ratio, the greater the rate of diffusion, and the vice versa is true. That is, the lower the ratio, the lower the rate of diffusion. Now this implies that a small organism always expose a large surface area to the surrounding when compared to big organisms. So we have a table, and we'll compare the volume ratios of different organisms depending on size. So we look at the relationship between surface area and volume ratio. So I've just mentioned, just as I've mentioned earlier, that different organisms have different surface area and volume that is always exposed to the surrounding. Now I have different objects here. So we have this one having a cuboid having a measurement of 1 centimeter. The other one is having 2 centimeters. The other one 3 centimeters, 6 centimeters, and 12 centimeters. Now, if you calculate the surface area for this one, it will be 6 square centimeters. This one will be 24 square centimeters, 54 square centimeters, 216 square centimeters, 864 square centimeters. Then you can also calculate the volume. So you'll find that for the first one, it will have a volume of 1 cubic centimeter. The other one, 8 centimeters, 8 cubic centimeters. This other case, we have 27 cubic centimeters, and the rest. So what you need to realize is that we are just trying to compare the ratio of different organisms depending on the size. So we said that, or we had mentioned earlier, that the higher the ratio, the greater the rate of diffusion. So from what we have, you are in a position to tell where we have the rate of diffusion being greatest. So just look at the ratio, the ratio of the ratio the volume ratio of the organism depending on the size. Now you realize that because small organisms always have larger ratios compared to large organisms, the small organisms will always depend majorly on diffusion as a means of transportation of foods, respiratory gases, and waste materials. But for the case of large animals, in as much as we also have diffusion used as a means of transport, but they have to have another transport system which ensures that the materials are transported throughout the body. So that one marks the end of the lesson. Next lesson we will be looking at the last thing on diffusion, and that will be the role of diffusion in living organisms. Thank you. I hope you enjoyed the lesson. Let us meet next time.